Okay, it looks like it's top of the hour. Thank you everyone for joining us today for our webinar. Just a couple quick things before we get started. You can submit your questions via the questions panel throughout the webinar, and we will get to those at the end. And your certificates will be available this Friday by logging into your account. Dr. Meier, if you're ready to get started, go right ahead.
I'm ready. Thank you, Kim. So it's always exciting to have the opportunity to do one of these webinars. And I'm particularly excited today because I do have some new data that I'll be sharing with you for the first time. So the focus of today's presentation is improving mother's own milk feeding rates at NICU discharge. And the way we're going to talk about doing that is optimizing the achievement of secretory activation and coming to volume as key strategies for achieving that end goal. So first of all, just for purposes of disclosure, I've listed the research and demonstration projects, as well as the fact that I have received payment for presentations and consultation from the DILA in the past and continue to do so. So as far as presentation objectives today, we'll first start off by citing evidence just very briefly about differences in outcomes for mother's own milk-fed versus donor human milk-fed premature infants, because this is what we have to have in mind as we underscore the prioritization of acquiring mother's own milk and hence the investment in the infrastructure that is necessary for doing that. Second, we'll define relevant terminology. And as we do this, we're going to go through kind of a trajectory of how each of these things happen, looking at the evidence for each one. And then finally, 3 and 4, we're going to focus on the implications for clinical practice with respect to understanding the data and how we put those into best practices and protocols. So let's look quickly at the evidence and differences. So our data that I've summarized here from the Love Mom cohort showed that high dose mother's own milk reduces all of the different complications and morbidities and their associated costs when compared to formula. And this was with our cohort of 430 infants. I've summarized all the different papers on this topic that go all the way from 2013 to our most recent one just last year that was in pharmacoeconomics that was really looking at the cost savings of mother's own milk for this population. However, no donor milk was used in this cohort. This was all mother's own milk versus formula. What we do know with mother's own milk compared to either formula or to donor human milk is that increasingly it's being looked at as a personalized medicine, because the mother personalizes so much of what is in the milk for her recipient infant. The taste, the smell, the microbiota, mucosal immunity to microbes, and that is enhanced when mothers handle their infants and hold their infants skin to skin, for example, in newborn intensive care. Tolerance to environmental allergens, and I think most importantly, where the research in this direction is going, is that mother's own milk doesn't just help to stimulate and support the growth of these developing organs that are so immature at birth. It protects them while they are developing from all of the NICU stressors, such as oxidative stress, poor nutrition, and inflammation. Now, just again, because I want to clarify, mother's own milk isn't the same as donor human milk, which I think people realize in principle, but there are some new evidence that really highlight quite a bit of difference between mother's own milk and donor human milk. So just in terms of nomenclature, when milk from the mother herself is either fresh or frozen and fortified, it reduces prematurity-specific complications and their costs in a dose-response manner, with the highest dose promoting the most cost savings and the most beneficial outcomes. So high dose mother's own milk yields the best reduction in risk for all of the different complications and their cost. However, these beneficial outcomes don't extend to pasteurized mother's own milk, And in fact, the studies of pasteurized mother's own milk really suggest it's not that much different from donor human milk. So I think this is an important thing to keep in mind. Donor human milk reduces necrotizing enterocolitis basically in every study that has ever been conducted, but it doesn't really have an impact on any of the other complications or morbidities that mother's own milk is protective against. Now, this is where I want to focus our time here for the next couple of minutes. And this is what we call human milk or exclusive human milk feedings. Because human milk connotes that there are combinations of mother's own milk and donor human milk in the cohort of infants that are studied. But the concern is that many people that write about donor milk infer many of the benefits of mother's own milk into populations of either human milk-fed infants or even to donor human milk-fed infants. So this is where the new research is taking place, and I want to share that with you. But the other then final one is exclusive human milk diet, which usually means that there is a combination of mother's own milk, donor human milk, and human fortifier as opposed to bovine fortifier. So instead of going through a number of different donor milk studies, since that's not really the focus of what we're doing here, what we're doing here is to understand that there is a unique role for mother's own milk, and that's why we have to prioritize it through the different practices I want to share with you. But the emerging evidence about exclusive human milk diets, so keep in mind, these are people that publish about the fact that babies receive both mother's own milk and donor human milk. There are now 3 studies that I've referenced here, and I was actually invited to write the commentary that you see cited here for Cell Reports Medicine as a result of one of the papers that they had published, which is the Kumbar. So what these studies all showed was instead of having any formula whatsoever, these were infants that were human milk fed. But what these researchers did was to tease out what proportion of mother's own milk versus donor human milk infants received. So for example, in one of the cohorts that talked about exclusively human milk fed infants, divided the infants into the top 50 and lower 50 percentile based on how much mother's own milk they received. So if you think, okay, well, that's half and half. Well, the top 50% received on average 91% mother's own milk, whereas the lower 50% received on average 14% mother's own milk throughout the NICU hospitalization. So those 2 feeding groups were combined and are combined in studies we read all the time with respect to the fact that they were looked at as the same because they were all exclusively human milk fed. So the commentary summarizes briefly what the primary issues are, as you see here. And what those studies all showed is when the amount of mother's own milk fed is compared with the babies that get primarily donor milk or a lesser dose, is that there are significantly different outcomes for the infants. So babies who receive primarily mother's own milk have significantly better clinical outcomes, as well as metabolic outcomes in terms of gut microbiome colonization, as well as different metabolic pathways that are triggered by the 2 types of milks. So what I have as a final commentary in this overall paper is that we really have to prioritize mother's own milk, meaning that barriers to mother's own milk feeding really have to be met with the same economic support as often put into acquiring pasteurized donor human milk. So the reason I went into this is because there is a slippery slope. And this is often when you read in a paper, well, when mother's own milk is not available, whoever it is recommends donor human milk. Donor human milk is the next best thing. Donor human milk is appropriate when mother's own milk isn't available. And this is such a slippery slope because the real question should be, why is mother's own milk unavailable? Why are we not feeding babies a higher dose of mother's own milk? Because I've often argued that donor human milk should not be the solution for the lack of evidence-based best practices that increase the use of mother's own milk in the NICU. And the reason I started with this as an introduction is because we have to first understand that mother's own milk isn't easily substituted and should not be substituted with the next best option, which is donor human milk, because the difference between the two is so very striking, and it's beginning to appear in much of the new evidence in this area. So how do we acquire mother's own milk? Where do we begin? Well, I want to go through milk production and secretion just from the perspective of what happens during this critical period of time that mothers first give birth and begin to establish a milk supply. What we know is that throughout the world, the continued provision of mother's own milk for premature babies remains a problem, even though the rates of initiation are very high. In many places in the world, the rates of initiation of milk provision for mothers of premature infants is higher than mothers of healthy term infants. But what everybody struggles with is those same rates don't carry through to NICU discharge. Now, the primary reason for this, if we look at all the literature, is that mothers run out of milk. There's insufficient amounts of milk. And also, if we look at the literature, most people say, well, this timing usually coincides as the infant recovers and needs more milk, and the mothers don't have available milk. So if we look at what all of the data show at this point, and what's different when you have a premature baby in the NICU. So we've been publishing now really since 2015 that our work, as well as that of others previously, indicates that the first 2 weeks postpartum is a critical window with respect to long-term lactation outcomes. And this isn't just with premature infants, this is with healthy full-term infants as well. But our data specifically looked at a benchmark that we've for years called coming to volume, or achieving 500 milliliters of pumped milk each day by 14 days postpartum. And what our data showed in our sample of 430 mothers of the Love Mom cohort was that this was the primary predictor of mother's own milk at discharge. It was a bigger predictor than race, than ethnicity, than income, than gestational age, was whether mothers reached 500 milliliters a day for by the end of 14 days. This predicted going home, getting milk. So then that takes us back a step and say, well, let's look at the first 14 days in the life of a baby in the NICU, particularly a baby who's premature, or even a term baby who's in PO or very small amounts of milk. There is a mismatch between the amount of milk the baby needs for full feedings versus the volume of milk that's needed to regulate lactation processes. This creates confusion for families and staff. So if we look at milk removal, people often just think of milk removal as important for the baby to have intake, which is true. It is important that it assures adequate intake, but milk removal and the processes involved in it also regulate lactation and send messages back to the brain and to the breast tissue, so that what we have is two outcomes of milk removal. So if we talk about removing enough for the baby in the NICU, the baby might need as little as 100 milliliters a day on day of life 14, if it's a small baby or a baby who's fluid restricted for some reason. And the mother thinks, okay, well, I'm making 200 milliliters of milk a day, so I'm really doing just fine. However, the data tell us that to be able to continue lactation after this period of time, the mother needs to produce 500 milliliters of milk a day, even though that might be considerably more than the baby's able to take. So this mismatch creates a lot of confusion on the part of the staff with respect to teaching mothers, and for mothers who are getting most of their information from websites or from people who don't work with pump-dependent mothers of babies in the NICU. So let's take a look here at this critical 14-day window, and we're going to break this down as we go through. So there are implications for lactation that start pre-birth, which would be, as we talk about definitions, secretory differentiation. Then at birth, what happens is that with the birth of the placenta, the progesterone falls because the placenta has been secreting progesterone. And that's what we see here. We see that the progesterone plummets, and that is the trigger to unleash the milk synthesis role of prolactin. So that is the next thing that has to happen. And you'll hear us talk about many times that the initiation of lactation is a one-time event. It's one time because this doesn't happen more than once. We see the placenta delivered one time. So that's where the nomenclature comes, that it is a once in a life event. So the next 1 to 2 days, again, what we see is this fall in progesterone that triggers lactation. The next 2 to 3 days, if we look at the literature, is when we see secretory activation or the milk coming in. Day of life 14 is the next benchmark for 500 milliliters of milk. And then going home at NICU discharge, still providing milk for your baby, is the outcome we want to get to. So what I want to do in my next set of slides is to go through these processes, define them, how we measure them, to accomplish objective number 2. Okay, so let's take a peek at what happens during secretory differentiation during pregnancy. Well, we have to kind of keep in mind that the breast is under the same control as the pregnancy hormones. And during this period of time, what we see is that there is tremendous growth in branching between the secretory cells, the ductal branching, but we know that the milk synthesis itself is held in check by circulating progesterone. And as we will come to the next slide, there is increasing evidence that this process, the secretory differentiation, in other words, the growth and the development of the breast, making it capable of lactation, is affected by inflammatory and metabolic complications that may make lactation impaired as opposed to simply delayed. At birth, what we see is the placenta is delivered, and the placenta delivery triggers lactation in all mammals, not just humans. And then secretory activation, or milk coming in. What we see with this is it's a very complicated process. It's an upregulation of 4 transcellular pathways that synthesize milk components, the closure of a single paracellular pathway throughout the gland, and there seems to be increasing evidence that stimulation by the infant or the pump is really essential to catalyze secretory activation. But before we move on, we talked about secretory differentiation, or the preparation of the gland for feeding. So we have a fair amount of research right now in the animal work that shows that inflammatory processes, and for the most part, these have been studied primarily with overweight and obese animals because those conditions can be induced in the laboratory, that those conditions in the mammary gland during pregnancy affect gene expression for prolactin signaling and for receptors, among other mechanisms. Well, what we're concerned about, the group of us who's doing research in this area and trying to determine how do we apply these data to NICU mothers, are concerned because many mothers of babies who are in the NICU, especially born prematurely, have multiple inflammation-based complications: preeclampsia, overweight and obesity, diabetes, as well as incomplete mammary epithelial development, and then pump dependency. So the research priorities, I think, moving forward is trying to distinguish in this at-risk population between delayed and impaired lactation. Like, when do we know that a mother's done everything she can and the underlying problem may be secretory differentiation in the presence of these morbidities? And secondly, can we develop strategies post-birth that can at least partially mitigate some of the disordered secretory differentiation? So as we think about what happens after birth, I don't want us to lose sight at this point in time that a lot of the work right now is focused in certainly in animal models because of the ease of study, but focusing on what might be happening during in mothers who have inflammatory or metabolic disorders. Okay, so let's look at now what happens from birth to secretory activation achievement. So at birth, the placenta is born. And then that triggers over the next 1 to 2 days this precipitous drop in progesterone. Okay, well, what do we know about that? So let's take a minute to peek at that process. One of the things we know is that that is affected, at least a mechanism is thought to be affected by adiposity because fat tissue harbors progesterone. So instead of seeing the progesterone excreted via the urine in a 2 to 3 day period, we could see prolonged progesterone in mothers who have high BMI, overweight, and obesity. So that would explain, at least in part, some of that delay in secretory activation. We obviously know about retained placental fragments. And for those of us who still remain concerned about the use of early hormonal contraception in this population, this is also the concern, is because those remedies or the contraceptive active ingredient are all progesterone based. So it's putting progesterone into the mother's circulation at a time that it needs to fall so that prolactin can synthesize milk. So this has been the concern all along with early hormonal contraception, is its timing interfering with the decline in progesterone. So then as we move to secretory activation, well, interestingly, again, the literature shows 2 to 3 days for healthy mothers and babies, and lactation secretory activation as measured by maternal perception in most studies is thought to be delayed if it occurs after 72 hours. Well, what our new data show, which I'm going to share with you, is that this process is affected by pumping frequency. We assume and think it may be affected by ineffective pumping pressures, shield fit and placement. In other words, the mother's pumping, but isn't really effectively stimulating the gland. We know it's affected by maternal overweight and obesity. And we think, based on clinical reports, that it may very well be affected by other comorbidities and that may mask disordered secretory differentiation. And in fact, our most recent study called Making Pumping Better for NICU Moms, which I'm going to discuss in a moment, showed that the average onset of secretory activation in this high-risk population was 5.8 days postpartum. And we didn't have a single mother achieve secretory activation before day 4. And this was by measuring secretory activation using milk biomarkers, which I'll discuss. So we are definitely, what we've seen in all the clinical work, that it seems to be delayed. It is delayed. And I think just having this information to share with mothers, I know myself to be able to say to a mother, well, the average for this is 5.8 days, as opposed to saying, well, we need your milk on day 2 because your baby needs to be fed. We need your milk on day 3, as opposed to having mothers understand that this delay is normal. So what happens during this period of time? What is the physiology? Well, what we do know is that when mothers give birth to healthy full-term babies and the babies breastfeed, they're not separated from the mothers whatsoever, and they're weighed before and after every single feeding over that 24-hour period of time, that the average healthy infant consumes 15 milliliters of colostrum in the entire first 24 hours of life. This is 1.5 milliliters per feeding, and the average baby fed between 10 and 11 times. So what happens on day 1, that this is the norm in a healthy mom and baby, all the way through to day 4 to 7, when mothers of healthy babies are already achieving what we would call coming to volume. We're making all this milk. So this is, it's almost like you look at it and think, this is just a miracle. And I guess I promised myself I wouldn't get dissuaded as I talk to you today, but I get so excited sometimes with examples. But I think one of the best things we can say to a mother who's pumping drops of milk over this first day or so, I always love to share that it isn't like a little bit more each day. It's like all of a sudden the breast turns on and we see a change from half a teaspoon of milk to a cup of milk over the course of a 24-hour period. And I think that that's something really important because mothers don't know. They think it's going to be these little drops all the time. So what's happening at the level of the mammary gland? And I want to explain this because this is the basis for the new measures of secretory activation I want to discuss, which is using milk samples to be able to tell when a mother has achieved secretory activation. So if we take a look here at this diagram, these are the individual cells in the breast that synthesize milk. So milk would come from those cells into the duct, this is the duct, the lumen of the duct. And this would be the interstitial space. So the mother's milk supply or the mother's blood supply is providing the nutrients, providing the energy so that the breast is able to synthesize the milk. This is what the breast looks like before secretory activation, and this is after. So let's take it in. I got it coded, so pink. So one of the main things that happens is before the mother actually experiences secretory activation, there are spaces between these individual milk-making cells. Those are called paracellular pathways. And what happens in lactation, because the mother's bloodstream and the milk, and the milk can actually interchange over this period of time, is that based on osmotic gradients, components that are made by the breast in the form of milk products can pass back into the maternal circulation, and things that are in the maternal circulation can find their way into the milk, that after full lactation, secretory activation, and we see the paracellular pathways close, that channel of communication is closed. So this is why, so many, we talk about the blood-breast barrier, because so many times a medication that a mother is taking, we talk about, well, the medication can't get into the milk, even though it's in the mother's bloodstream, because these paracellular pathways are closed. So if it's a high molecular weight medication, it can't find its way into the gland. So it's the same principle here. So what happens as the transcellular pathways are upregulated, this is what makes the milk components, is lactose is made in transcellular pathway number 1. Okay, so it's made, and ordinarily it would stay in the gland and become the substrate for the infant to remove. However, during early lactation, when these paracellular pathways are open, lactose goes back into the maternal circulation and is excreted in the urine. In contrast, sodium comes from the mother's circulation and goes into the milk. So what we would see is during early lactation, if we wanted to take a sample of the mother's milk, when the paracellular pathways are open, we would see low lactose and we would see high sodium. And for those of you who are thinking along the lines of colostrum, we would also see high total protein, because the mother's, all of the different components that are the protective proteins from the mother pass through these open paracellular pathways as well and make up what we call colostrum. Now, once we see secretory activation, these paracellular pathways close. Lactose stays in the gland, and sodium comes only from the cell in the epithelium. It doesn't come from the mother's circulation. This is important because lactose is the primary osmotic driver of milk supply. And as long as it is going back out of the breast into the mother's circulation, a full milk supply is not possible. So as we talk about which biomarkers to measure, some people think it's redundant to measure both lactose and sodium because they reflect open paracellular pathways at this period of time. So the sodium is usually the marker, because once we see the paracellular pathways close, the sodium in the milk goes down. So this is actually a kind of a good example right here. It's taken from my PhD student, Clarissa Medina Polenis, who's soon to be Dr. Medina Polenis in another month or so. If we look at birth and then these changes as secretory activation, what we see is that the sodium and the protein come down, again, because of the paracellular pathway closure. The lactose, the citrate, and potassium increase over this period of time. And when we look at what would be, what many people would refer to as normal values of milk biomarkers, so if we're collecting milk and we say, has the mother achieved secretory activation? It's usually sodium that's under 16 millimoles, or if we're using the ratio of sodium potassium, which is kind of a way of standardizing sodium, which many people think is probably not additive to sodium potassium ratios, or to sodium alone, that ratio is usually considered to be under 0.8. And this is what would be used in most instances to say, okay, the mother has achieved secretory activation. But equally telling for practice is going to be the fact that these trends are also really, really critical. Because even though, let's say we've got a mother on day 3, and even though she may not have achieved a sodium of under 16, her sodium has decreased markedly over the last 3 days. It may not be 16 yet, but we're seeing that trend in the right direction. So there's a lot of room in this work, not just for values, but for trends in the milk biomarkers in the early post-birth period to examine secretory activation. Okay, so we've talked about this. How easy is it to do? It involves taking a single milk sample. In our work, we've measured these either daily or twice daily to look for the change in either sodium or sodium potassium ratio. It involves quite simply taking a sample of the milk, putting it on one of these little electrodes, and interpreting the results. Now, this is a new field, but it is nonetheless one that's here to stay. Our team is working quite a bit on this, and I've joined forces. Dr. Leslie Parker, many of you know, and I and our various colleagues and students and Dr. Hoban have begun to look at this phenomenon in mothers with babies in the NICU. So there are several papers right now that have reported biomarkers of secretory activation in at-risk mothers. And if anyone is interested in the actual papers, they're here, although I will be talking about a couple of them as we go through. So in terms of further defining terminology, I want to share with you some data out of our study that we called Making Pumping Better for NICU Moms. So this was a 14-day observational study. We collected measures every single day on our mothers. We enrolled 35 mothers, and we have 29 with usable data. What made this study so interesting was it gave us the most accurate possible measures of pumping behaviors that have ever been recorded thus far, because we worked with Medela engineers to design what we call a smart pump that electronically measured, recorded, and stored when a mother was or wasn't pumping. So we have every minute of pumping that the mother engaged in over the first 14 days. So each mother had access to 3 pumps, one in the maternity setting. So when she had just given birth for those first 2 to 3 days, had one in her room, one in the NICU, and one to take home. So we were able to capture virtually all of the pumpings over this 14-day period. Not only that, but we were also able to work and create an app that allowed the mother to weigh her milk. That's what this little scale is here. So the mother was actually able to weigh the milk separately from the right and the left breast to the nearest 1/10 of a gram, so that we had, for that particular milk pumping session, exactly how much milk the mother had removed. And we also then collected a variety of other things in terms of breast photos and scans, maternal descriptions of breast changes, with the hope of trying to validate some of the milk biomarkers with maternal perceptions of milk coming in. And these will be things that my student, who will be Dr. Medina Polinas, will be looking at as her first work. So I want to share with you the data from this particular study as we go through again with further definitions of secretory activation and using the biomarkers and coming to volume. So the first thing we looked at is we asked our statisticians to do a cluster analysis for us based on pumping behaviors. We said we're interested in looking at how many minutes a day the mother pumped. We're interested in looking at how many daily number of pumpings, and what was the longest interval between pumping. Well, these were all very tightly correlated, and as you can see, they fit nicely into two patterns of mothers. Group A were the high pumpers, and Group B were the mothers who pumped less frequently. And by I mean less frequently, less minutes, so they had fewer minutes that they pumped. They had fewer pumping sessions than the other mothers, and they also had longer intervals between pumping for the entire first 14 days. So it's with this cluster analysis that then we began to look at, okay, well, who achieved secretory activation? Who achieved coming to volume? What was that trajectory? And because these were all so tightly correlated, the one that was the most, I guess I would use the word stable, that we chose to work with was daily number of pumpings. So if we look at pumping. Okay, one of the reasons we're interested in this is because it's modifiable. Okay, a mother's BMI may not be modifiable. A mother's fact that she's had a cesarean delivery and a 25-week baby isn't modifiable, but pumping is modifiable. So let's look first of all as how did pumping impact the achievement of secretory activation? So here along this graph, this is daily sodium, this is days post-birth. I drew in here 16, okay, millimeters. This is when we say the mothers achieved secretory activation. So what you see here, the mothers who pumped more frequently, these were the mothers in cluster A. 94% of those mothers achieved secretory activation. And as you can see, as a mean, they did so again between days 5 and 6, okay, and stayed there. If we look at mean values, the mean values for the cohort of mothers, the cluster of mothers that pumped less frequently, the mean value never did achieve secretory activation, although individually, 58% of those mothers did. So that was a big difference. But again, some of these I just can't wait to put into talking points, because the odds of a mother achieving secretory activation in the next 2 days increased by 48% for each additional pumping session. So one more pumping session and you increase the odds that you'll achieve secretory activation by 48% in the next 2 days. So if we look, as you can see, and this is one of the things when we talk about personalized intervention, this is looking at each one of the mothers separately. Okay, so I know it's messy, it's 29 mothers, but it is looking at the number of pumping sessions and achievement of secretory activation. And what I'm going to point out here, again, although there's tremendous variability, is that we found out for the first time with this that secretory activation really wasn't permanent. I shouldn't say the first time. We had inklings of this from Dr. Hoban's 2018 study, but this was even more profound because what we found from this, I'm going to skip ahead here for a second, when I talk about impermanence, was that over half of the mothers in this study that achieved secretory activation at some point during this 14 days reversed it at least one point, reversed it at least one time in the first 14 days. So in other words, the sodium came down below a 0.8 and then something happened and it went back up. Now this reversal was almost always due to an interruption in pumping frequency. And we have here, I'm actually going to go back now. I'm sorry for the confusion, but this just made sense to focus on it right now. The reversal was almost always because a mother had skipped a pumping session or pumping sessions. So now let's look at what we just focused on with respect to the pumping and its relationship to secretory activation. And we look at the fact that more pumpings in this first 5 days really indicates that the mothers are going to make more milk. So this is an actual set of data from a single mother, and it represents sort of what is the trajectory for a mother who pumps effectively, efficiently, and this would be what I would call the norm. So this is looking at how much milk was removed. This is looking at postpartum day, and this is looking at hours pumped. So in pink, we have how many minutes the mother pumped, or I guess that's how many hours the mother pumped in this graph. and blue is how much milk was removed. So what this means is in this early period of time, mothers really have to trust you as a clinician saying to them, it'll get better because you are pumping a lot and you're not getting that much milk. Because by day anywhere between 6 and 7, we begin to see this reversing. And the mothers are able to pump fewer times and make more milk. So the early pumping in this early 5 to 6, 7 days seems to be really, really critical for these mothers. And as you can see, this is the sodium potassium ratio, and the mother with the continued pumping, we see it falling, and it's below 0.8, almost here, but below 0.8 on day 6. So again, this whole period of time between 5 and 6 for achievement of secretory activation. Now, on the other hand, I want to go to the bottom graph. I talked about this one and the lack of permanence, because this is, again, a sample from the same group of mothers. This was a mother who had tremendous comorbidities that included retinal detachment as a part of her preeclampsia and high blood pressure. So what happened here, this mother, again, as you can see, was pumping frequently. And then this was the day, and she was almost had achieved secretory activation, but this was the day she had to go back to the ophthalmologist and was in and out of procedures all day long. And what happened was, as you can see, what was almost an achievement of secretory activation reversed itself. And she had pumped just one time the day before. It reversed itself. But as she increased her pumping frequency, what we see is that she once again achieved secretory activation and was able to remove more milk with respect to how many minutes she was pumping. I bring this up because this is really something that our bedside nurses were the ones that usually picked up. It was that a mother wasn't pumping frequently enough, or a bedside nurse overhearing a mother saying that she just decided, this was another mother, decided her baby had enough milk and she was going to quit pumping on day 13. Well, the nurse said, you can't do that because the baby needs your milk. Well, the mother popped out of secretory activation and then into secretory activation in a single day because the nurse picked that up. Okay, so let's go now to the second, achievement of coming to volume and mother's own milk at NICU discharge. So what we know, again, 5.8 days is the average time that we began to see a mother initiate secretory or achieve secretory activation. What we know is that effective, efficient, and frequent milk removal during this period of time is essential. And these are some new data from the study. Each additional pumping hour, okay, so that again, an hour, in the first 5 days postpartum was associated with these day 7 outcomes. An additional 1,278 milliliters of cumulative milk volume. So that's not per day, that's over the course of the 7 days. And a 2.8 odds of sodium being within normal limits. So again, translating that to mothers, each additional pumping hour. And what does that mean? And then we know once we get to day 14 with having achieved coming to volume, that is the biggest predictor of milk volume going home from of milk, continued milk provision going home from the NICU. So we know that coming to volume is involved with a different set of regulation of lactation, because we change from hormonal to the autocrine and the paracrine regulation. And each breast operates separately, and we know that when milk is left behind, either because of ineffective pumping or long intervals between pumping, that it begins the weaning process. Now, two things control this. One is obviously frequency of pumping, because what we know with the frequency of pumping, as you see here, is that when pumping begins, the prolactin is released from the pituitary, and in early lactation actually triples its baseline concentration before it returns gradually to the baseline over approximately 2 to 3 hours. So the issue is one of this area under the curve is when milk synthesis is the most rapid, So this is why mothers must pump frequently over this period of time to be able to stimulate the release of prolactin. And as you can see, this is a graph that looks at over the period of time postpartum. So day 10, day 40, day 8, day 120, looking at prolactin. And this shows again, what is the baseline and what is the peak? What are we after we've stimulated the breast or the baby removes milk? This response is what is for continued lactation. But the main thing, it is the most disparate between the baseline and the peak in early lactation. So this first 10 days is when you get the strongest release of prolactin, so that you see the biggest pre-pumping to post-pumping difference. So the second mechanism is one of not leaving milk behind in the gland. Now, this is the mechanism for weaning. It's what we call supply and demand. But I would have you think about breast pump dependent mothers who pump infrequently or with ineffective or inefficient equipment inadvertently begin weaning. Okay, they begin weaning by the same mechanism. So it isn't necessarily intentional, but that's what's happening. So I would refer you to this magnificent paper that was a review of all of the different mechanisms by which milk left behind in the gland regulates milk synthesis and subsequent lactation outcomes. So it basically shows there's not just one thing, there are many things. But what I would have you keep in mind as working with this population is weaning can be inadvertent, and we should pick up the idea of mothers weaning when the milk supply starts to wane. So now let's go into practices. Okay, so I think the first thing we've begun to think about that I want to share with you is rethinking this critical window. Now, we've been talking about 14 days for some time. If we look at our data from Dr. Hoban in 2018 with the Love Mom cohort. However, what I want to have you think about here, this is the pumping clusters, again, from Making Pumping Better. And what we look at here is, again, the high pumpers are the mothers in red, okay, is that they continue to increase their rate of milk removal or milk volume, as we think about it, through about day 8, and then they plateau. And the same thing really, even though it was a slower rate of increase for the mothers who are less frequent pumpers, as you see here, okay? The same thing with sodium and with these two cohorts. So the sodium is decreasing more slowly in the mothers who pumped less frequently, decreasing most rapidly, having achieved secretory activation in the mothers pumping more frequently. But both groups, again, what we see as a plateau after 8 days. Well, that alone would have been interesting, but it makes us rethink this critical window because of some data that Clarissa Medina Polinas published in this paper in 2021. That was a sub-study or secondary analysis of Dr. Hoban's 2018 and 2021 papers on using the biomarkers. And she did a secondary analysis looking at milk volume and sodium over the first 14 days in mothers with a BMI that was equal to or greater than 27 and mothers with a BMI under 27. So looking at the effect of maternal BMI. And what she showed, again, days postpartum 1 through 14, what we see is that the mothers who were normal BMI, so less than 27, had a significantly greater rate of increase in milk over that first 7 days. And that was 65 milliliters per day increase on average. In contrast, the mothers with the higher BMI were making on average 33.1 milliliters a day increase. But again, what happened at the end of the 14 days, or I'm sorry, at the end of the 7 days, is that these differences basically plateaued. There was still a difference between the two groups, but there was no greater milk yield in either group. It was a plateau. So this makes us think, okay, what is our window of intervention? 7 days. The same thing with with Clarissa Medina Polena's data on sodium. As we look again, what we saw was a significantly rapid decrease in the mothers um, who were lower BMI. So we saw a significantly more rapid decrease in sodium, which we would expect because that signals secretory activation. But at the same time, we saw a slower one in the mothers with higher BMI, but after that 7 to 8 days, not much happened. So this makes us then rethink, okay, is our critical window less than 14 days? And we'll be doing some studies to examine that. But I think the two studies that we have make us rethink that it may be a shorter window with respect to getting the mothers off to the best start using the physiology of lactation during that time. So I want to end with looking at best practices, because everyone says, okay, but what about hand expression? Well, I think the important thing is that there really are no data whatsoever to inform the use of hand expression as a substitute for breast pump use in mothers of preterm infants. Nonetheless, it's often recommended. In fact, there are some instances where I've been told mothers of premature babies have to be taught how to do hand expression before they can leave the maternity hospitalization. And this is really concerning when you think about the mothers, because everything tells us how much stress they're in, how much difficulty they have making decisions and learning. And the pump itself is complicated enough without teaching them something for which there really doesn't seem to be evidence at this point in time. Now, if we were to do studies to look at the effectiveness of hand expression, based on what we know now from secretory activation and coming to volume and this trajectory I've spent all this time talking about, we can't look at individual milk removed over as a mother begins to transition from birth to secretory activation. These are small volumes, and that isn't really the issue. The issue is, does the mother have greater cumulative volume at the end of 7 days, at the end of 14 days? Does she initiate secretory activation comparably? We don't have any data about that from hand expression, but we have those data from breast pump usage. So let's just kind of go through here. What has been some of the study in this area? We have 2 studies that have looked at breast pump versus hand expression in the early post-birth period. And I'm going to not go into each one of them, but I know that this one is sometimes used as the rationale for hand expression. And what we know is that in this one, mothers alternated hand expression with pump use, which is an unsuitable research design because one is going to influence the other. You can't compare what happened at 4 a.m., at 6 a.m., just depending on whether the mother was hand pumping or hand expressing or pumping. The mothers reported less discomfort with pumping, but the investigators still recommended hand expression. This, I think, is the most telling study, and in the absence of another study that conflicts with it, I could not recommend, or I think it would be unethical at this point in time to almost do a study like this where we already have pumping available. If you're in a country where you could randomly assign pumping and hand expression ethically, that might be different. But this particular study assigned mothers randomly to exclusively use an electric pump or hand expression for the first 7 days post-birth. The mothers were not different in their characteristics. So after the first 7 days, the mothers who were using hand expression changed to pump use, but they never caught up in terms of milk supply, either over the course of a day or over the course of cumulative pumping. So that the electric breast pump use in the study was associated with an additional almost 120 milliliters of milk per day over the first 28 days post-birth when compared with hand expression mothers. Again, this suggests a type of programming, because if the mothers switched to the pump at 7 days, but still were not able to make comparable amounts of volume, really suggests that we miss this window of programming by not stimulating the breast appropriately. So we're often told, I really dug through the old history here because I wanted to just kind of put some of these questions that I've been ask about to rest. One says, oh, well, you get more volume when you use the hand. Well, this was a study, and we probably couldn't even do this study today in terms of how well controlled it was, because these researchers not only measured volume using different types of milk removal, they also measured this prolactin curve that I was talking about. So if we look here, the hand expression was the least effective in removing milk compared to all of the other pumps. We look here, and I've often heard, oh, people say to me, well, the prolactin is supposedly like when the baby feeds. Well, that wasn't the case here. The prolactin was like half of what it was when the baby fed. So we don't really have evidence for prolactin. Here again is an older study from 1996 with subjects randomly assigned here again to milk expression using the hand and using the pump. And what you see here is, again, total milk expressed was almost just half of what it was with the pump. This was a randomized study that I consulted with Dr. Slusher in nurseries in Africa, where mothers were randomly assigned to use an electric pump, a pedal pump, and hand expression. So obviously the electric pump was superior because obviously it's more efficient, but even the pedal pump, which is the same thing, it uses suction, it isn't expression, uses suction, was at least suitable in terms of milk removal for this population. So what I've talked about with hands only expression is not the same as hands-on pumping. This was Dr. Morton's work, but even at that, her work has been interpreted many times to mean that you don't need a pump. It's also sometimes been interpreted to mean that a mother really needs to do hands-on pumping. And I don't want to in any way, if you as a professional feel a mother would benefit from additional hands-on pumping or additional expression in addition to the pump, then I think that's your decision as a clinician. You must do that. I think the data tell us we can't substitute. But if we simply are using data to say, well, Dr. Morton's study said we need to do this, what I'd have you look at is Dr. Morton had 3 groups of mothers, and it was not a random assignment. These were mothers that were self-selected based on how often they decided to do hands-on pumping. So group 1 that you see here did the least hands-on pumping. Group 2 did more, and group 3 did the most. But if you look closely at the data, those mothers also each used more breast pumping. So the highest group of mothers that did the hands-on pumping also pumped more frequently. So you can't really sort out this is hands-on pumping versus just stimulation of the breast from the pump. But again, I think that's not the battle. The battle is not using just hand expression. So I would point out again, another best practice is to implement the Provide Compendium toolkit. And there are so many beautiful videos in here that tell you and show mothers how to use the pump appropriately for shield placement, pressures, breast shield sizing, pumping to empty. And just as a final thought, to optimize pumping, we're going to have to move towards optimizing pumping by standardizing certain procedures that I've listed here. Most of the time, or I shouldn't say most, many times mothers are just given a pump and saying, have at it, go to it. They get their information from websites as opposed to people that are experts. I think future, we're going to see personalized pumping strategies. We're going to optimize the pumping for everybody. And then we're going to use daily biomarkers to share the progress with mothers. I think it's important to determine how much of this mothers can do on their own, because I think they would be empowered with this. I think there are multiple quality improvement opportunities along with this to share in this science with mothers so that they personally can see the trajectory and get feedback on their pumping behaviors. So I'm not going to read the summary. I know we're close to being out of time, but what I want to leave you with is right now all of our studies are showing that the pumping behaviors are the primary predictor of achievement of secretory activation and coming to volume. I think the other important thing is that mothers need to be encouraged that if they don't see copious milk supply on day 3 or day 4, that the norm from our studies is now looking that it's between 5 and 6 days, and this is the norm. So I'm going to leave it here and turn it back to Pam.
Thank you, Dr. Meier. That's okay. Thank you, Dr. Meier, for that presentation. We do have maybe a minute for a question or two. I'll get right to that. The first question that came in is, what is the normal average amount of milk mothers produce per day for a 32-week preemie at 2 weeks of age?
Okay, well, I guess the answer to that is what would be the average amount. I hope what I've shared with you here is that that is modifiable. I mean, that it looks to be modifiable based upon pumping behaviors. So certainly at 32 weeks gestation, at 14 days, there's no reason, unless there's an underlying serious comorbidity, that a mother shouldn't have 500 milliliters a day. I've seen mothers with 1,000, with 1,500 milliliters a day, but certainly there should be the same threshold. It should be a minimum of 500.
Thank you. And it looks like we're already at the top of the hour. So again, thank you for that presentation. Just a couple quick things before we end. Just a reminder, your certificates will be available by logging into your account this Friday. And registration is open for the March 15th webinar. You can register and get more information at medilaeducation.com. And the evaluation will open up in a separate window once we end this webinar today. Thank you everyone for joining us today, and have a great rest of your day. Thank you.